So first of all, I'd like to thank Sarkis and Pramila for giving me this wonderful opportunity to present our data at this meeting. It's such an honor to me. So over the years, we've been interested in understanding how gut-residing bacteria affect immune cells in periphery, and how immune cells in periphery also affect animal behavior and brain development. So for example, recently we identified a derivative of bile acid, which are microbial metabolite, and we found them as immune modulators in controlling differentiation of inflammatory and also anti-inflammatory immune cells in the lamina propria of the gut mucosa. And we also been working on the role of immune cells in controlling animal behavior again, and then we've been particularly focused on the role of a soluble factor, cytokine, that produced by immune cells. And we've been working on this, their function in affecting brain development during the embryogenesis and also affecting neural activity in adult mice. So previously, we showed that this inflammation and immune cells in pregnant mothers has ability to affect this fetal brain development. And when this developing embryos exposed to heightened inflammation during the critical time window of pregnancy, they develop this behavior abnormalities or autism-like features later in their life. So according to human epidemiological studies, children born to mothers, pregnant women who are severely ill or hospitalized during the particularly late first trimester or second trimester of their pregnancy, and these children have shown to have a higher risk to develop neurodevelopmental disorders such as autism. So this observation that inflammation, exposure to inflammation in pregnant mother can lead to long-term behavior changes has been modeled in rodent, and which is shown here is called the MIA, or maternal immune activation model. So in this model, the adult offspring, particularly male mice born to these pregnant mice, which were exposed to inflammation induced by respiratory viral infection or injected with long double-stranded synthetic RNA, poly-IC, these offspring developed array of behavior symptoms, including social deficit and repetitive behaviors. So as a first speaker nicely illustrated, we often use this various social approach behavior as a way to measure their autism-like features in these mouse models. And in this case, a test mouse, when it was given a choice between social target, or which is a stranger mouse, or inanimate object, they tend to spend more time with the social target. However, male mouse offspring born to these pregnant mice that injected with poly-IC or infected with the viruses, they somehow lost interest toward the social target. And as a result, they spend a similar amount of time, or sometimes even more time, spend more time with the inanimate target. So by measuring this, the relative time spending with the social versus inanimate target, we are able to quantify their social approach behavior and compare the control animal, in this case, MI mice, again, mice exposed to inflammation during pregnancy, and later in their life, they show this deficit in social approach behavior. So using these animal models and various genetic and other immunological tools, my laboratory, also in collaboration with Gloria Choice Lab at MIT, Dan Limmat at NYU, and Chuck Hofer at University of Colorado, we have been able to identify key factors in this pathway. For example, we show that a subset of T helper cell, Th17 cells, and its cytokine, major cytokine interleukin 17A, play important role in the pregnant mother. And on the other hand, we show that a receptor for IL-17, the receptor that is expressed in the developing fetal brain, play a critical role in promoting this behavior abnormality in the offspring. We also show that introduction of a cytokine IL-17A into the fetal brain is sufficient to produce this behavior abnormality in the offspring. In addition, we show that this inflammation, maternal immune activation, only promote the development of this behavior abnormality when these pregnant mice carry a particular type of bacteria, gut commensal bacteria, in their intestine, which are known to promote the differentiation of gut residing Th17 cells. In other words, even if you induce inflammation in pregnant mice that do not carry this particular types of strain, bacterial strain in their gut, their offspring do not fail to show this behavior abnormality. So, we need to look at the inflammation. When we need to look at study inflammation, we also need to take into consideration of gut residing bacteria, community of gut residing bacteria in the pregnant mother. So all this data suggests that indeed the inflammation during pregnancy can promote this behavior abnormality in the offspring. So in a way, this inflammation can produce bad, not so great effect. It's not good to have this inflammation in this case. More recently, we also started to study or understand the beneficial role of inflammation, the kind of opposite phenotype that we saw in the previously. In other words, if we expose this adult offspring rather than developing embryos to the same type of inflammation in their adulthood, we actually now detect a mediated symptom in their behavior abnormality rather than promoting behavior abnormality. So this data illustrated the complex nature of the interplay that exists between host immune system and brain function and animal behavior. So this notion that inflammation, depending on the context, the inflammation or inflammation-associated fever can have a beneficial effect or can be used as a cure to treat various types of mental diseases is nothing new. So throughout the history, actually, there are people, like many philosophers in old days, also noted this phenomena. For example, both Hippocrates and Galen noted that the beneficial effect of fever on epilepsy or the beneficial effect of malaria-induced fever to treat this neuropsychiatric phenotypes. So this idea that the fever or inflammation can be used as a therapy to treat a psychiatric patient has been widely accepted up to early 20th century. For example, in 1927, Nobel Prize was given for the discovery of the therapeutic value of malaria inoculation in the treatment of this dementia paralytica type of mental psychosis. So basically, in this case, they took this blood taken from this patient, a malaria-infected patient, and then by injecting that blood into a patient with a mental illness, and that they were able to cure a certain percentage of those patients. On the other hand, some other patients eventually died due to the other complication. So from all this data suggests that this fever or fever-associated inflammation may have some beneficial effect in treating neuropsychiatric or neurodevelopmental disorder. More recently, this is a New York Times article that features this earlier work done by Dr. Curran and Dr. Zimmerman, and they look at this autistic kids, and they found that those autistic children are found develop this mitigated symptom when they are exposed to high fever. So this researcher noted that fever somehow restores some sort of a neural activity that do not work properly in autistic people. More recently, in 2018, a group of investigators studied with more than 2,000 autistic children, and they also found that 17% of these autistic children showed this improved behavior phenotype when they were exposed to fever or fever-associated inflammation. So based on all this earlier data and the suggestion, we decided to study whether our model can be used to help us understand the mechanism by which this fever or fever-associated inflammation can lead to this behavior rescue. So in order to expose our animal the mice to fever or fever-induced inflammation, we decided to use LPS, which is a bacterial cellular component, as a way to induce inflammation. So shown here on the left, mice were injected either with a beak or with the LPS, and as you can see, when we measure body temperature, LPS injection led to increase in the body temperature around 1 degree. And in this system, we ran behavior tests both with the control look at the MI offspring. As I told you earlier, we saw there's a reduction in sociability phenotype, and then vehicle treatment didn't really improve that. However, if we look at this MI offspring, before the LPS injection, they spend lesser amount of time with the social target. However, after LPS injection, we saw dramatic and robust rescue of their social phenotype, which lasts around 48 to 72 hours, suggesting that LPS-induced fever or inflammation has the ability to increase sociability phenotype in this offspring. So, and then next we like to ask whether this fever itself has the ability to induce the same phenotype. So to do so, we decide to induce the increased body temperature without inducing inflammation. So in this case, we express the inhibitory dread chemogenic opsin in the area of the hypothalamus, and by inhibiting this particular neuron with the designer drug CNO, we are able to increase body temperature. And in this case, again, these mice were not exposed to LPS-induced inflammation. And next, we ran behavior test, sociability test with this with a CNO injection. And as you can see here, CNO injection is not sufficient to induce social rescue, suggesting that at least in this condition, increase of body temperature alone is not sufficient to induce or increase sociability phenotype. So if it's not a fever, then what could be the mechanism by which LPS induce social rescue? So we next decide to look at the level of inflammatory cytokine produced in animal injected with a vehicle or LPS. As you can see here, compared to PBS offspring, our control group, MI offspring upon LPS injection are found with increased level of inflammatory cytokine, including interferon gamma, IL-6, and IL-17A. Among this, we decided to focus on IL-17, not only because IL-17, based on our earlier work, was shown to affect neurodevelopment and affect animal behavior later in their life, but also we found that there's a group of study, a group in UK reported this finding that IL-17 also in C. elegans can function as a neuromodulator, and they were able to show that IL-17 also in C. elegans can increase social aggregation phenotype in C. elegans by directly acting and modifying neural activity of the C. elegans. So based on this result, we next ask whether IL-17s play any roles in this LPS-induced social rescue. Again, when we look at this MI offspring, vehicle or vehicle LPS treatment with isotype LPS treatment to the mice that treated with those isotype antibody, we can see that there's a social improvement. However, if we inject mice with the LPS followed by anti-IL-17A blocking antibody into the ventricle, we no longer see that improvement. On the other hand, treating mice with anti-interferon gamma antibody, another cytokine that increased upon LPS injection, mice still display this improved sociability phenotype, suggesting that IL-17A but not interferon gamma in this model promote this increased sociability phenotype upon LPS injection. So the question is, what's the mechanism? So we want to understand how this LPS or LPS-induced IL-17 promoted sociability phenotype in this MI mouse model. Previously in 2017, we showed that in this MI model, we showed that there's number one, there's the increased neural activity in the sub-region of primary somatosensory cortex, which is a dysgranular zone of S1. And we also showed that suppressing of neural activity in this area is sufficient to restore sociability phenotype in MI offspring. So using this cortical layer specific neuronal staining and also using this CFOS staining, which is a surrogate marker for neural activity, we are able to at the time localize the area of brain shown here that carry a higher level of neural activity in the MI offspring compared to control offspring. So here you see many more green signal, suggesting that in MI offspring, S1DG area and also the surrounding region are found with increased neural activity. And we are able to also show that decreasing neural activity in the S1DG area using halorhodopsin, which is opsin to inhibit the neural activity, led to this social rescue phenotype, as shown here, compared to control group, suggesting that this suppression of neural activity in this particular brain area is sufficient to temporarily restore sociability deficit. And I don't have time to discuss all the other behavior assay that we ran, but also the same rescued phenotype was observed when we run other behavior assay, including marble bearing or open field test. So the question we asked was whether LPS-induced inflammation can lead to social rescue by affecting neural activity in the S1DG area of MI offspring. So we first look at this neural activity in the S1DG region with or without LPS treatment. Compared to control, in this area, we were able to detect again a higher level of TFOS activity in MI offspring, and we saw in there the reduced expression of TFOS activity in the animal that exposed to LPS-induced inflammation. The activity of LPS in suppressing increased neural activity is locus specific because if we look at other brain regions such as central amygdala, in this case, LPS actually induced or led to increased neural activity. Moreover, when we next measured the neural activity in live moving mice using multi-electrode recording on the S1DG region, and as you can see in this case, when you compare this vehicle-treated animal versus the LPS-treated animal, you are able to detect there's a decrease in neural activity in the S1DG area. And we know that this decrease of neural activity by LPS treatment is IL-17 dependent because the animal that treated with LPS followed by IL-17A blocking antibody treatment, we no longer see such a reduction in neural activity. So this thing that LPS has the ability to reduce neural activity in IL-17 dependent manner in the S1DG area of MI offspring. We next assess the expression of receptor for IL-17 adult mouse brain using in situ analysis. And as you can see, we saw a strong expression of IL-17 receptor A in the cortical region, particularly deeper layer of the cortex. So in order to test the function of IL-17 receptor in this LPS-induced rescue, we generated MI mice using IL-17 receptor flux line. And into these mice, in the S1DG of these mice, we introduced control versus Cre-encoding viruses. And then by doing so, we are able to deplete IL-17 receptor expression in the S1DG and the surrounding region. And using this animal, we subject them to LPS treatment. Compared to control animal, as you can see here, is mice that no longer have IL-17 receptor expression in the S1DG area respond to LPS, and then they no longer show this increased sociability phenotype, suggesting that IL-17A or LPS and IL-17A may increase the neural activity in the S1DG region by and then induce the social rescue by acting on IL-17 receptor expressed in the S1DG area. So we next ask whether this inflammation can also induce beneficial effect, not only in MI, but also in other monogenic mouse models for autism, such as CANNAP2, FLM1, or SHANK3. Since there are higher degree of mouse-to-mouse variability in these monogenic mutant mice, we first ran many mice with social tests and identified those animals that show robust social behavior phenotype. So we took these mice and subject them to LPS treatment. Again, in MI offspring compared to vehicle-treated group, LPS treatment, we saw this transient but improved social rescue phenotype. On the other hand, the same treatment failed to induce the social rescue in any of these monogenic mutants, suggesting that LPS-dependent behavior rescue may be MI-specific. However, when you look at the S1DG area of this monogenic mutant, we are able to find that they also have increase in neural activity measured by C4C expression compared to wild-type control. And also, this increase in neural activity in the S1DG area is functionally important because when we overexpress helilorhodopsin in that region and suppress neural activity, we are able to temporarily rescue social phenotype observed in both CANNEF FMR1 mice. So I think that similar neural pathway may play a role in contributing the social deficit phenotype in these monogenic mutant mice for autism. So next we asked then why LPS did not rescue the phenotype, social deficit phenotype in this mouse model. Clue came from this ELISA data. So we found that following LPS injection, there's an increased production of IL-17A in MI mice. However, we do not see such increase in any of these monogenic mutant mice. So next, we asked whether introducing IL-17A rather than injecting mice with this monogenic mutant with the LPS is sufficient to induce social rescue. Indeed, IL-17A infusion to S1DG brain area is sufficient to increase the sociability phenotype in this MI mouse model, and also both CANNEF2 and FMR1 show the sociability, mitigated sociability deficit phenotype following IL-17A injection. So from this data, I hope that I was able to convince you that immune activation and associated increase in the level of IL-17A is capable of rescuing this sociability deficit by directly acting on this S1DG brain area. And also, we are able to show that this effect of neuroimmune interaction can be generalized across both environmental and genetic mouse models for autism. So based on this data, going forward, we wanted to understand or develop ways to detect high-risk group of women who may have an increased chance to have children with a neurodevelopmental disorder during pregnancy. We also, if that's the case, if it's possible, we may be able to come up with a way to prevent the development of such neurodevelopmental disorders in their offspring by treating or by modulating the immune responses in the pregnant woman who are infected with respiratory viral infection. And this effort, I think, are very significant because I expect that unfortunately, due to this COVID-19 pandemic, there will be likely to be increased cases of children without ASD going forward because many pregnant women may be are infected with this SARS-CoV-2 virus. And lastly, using this immune system inflammation as potentially therapeutic tools, we are hoping to develop novel ways to treat this symptoms of neurodevelopmental disorder in the future. So having said that, so with this, I'd like to thank, many people who are involved in this project. Particularly, I'd like to thank Gloria Choi at MIT and her trainees, Mike and Youngshin, for their contribution to the work that I presented today. And essentially all of the work, most of the work that I present today were possible through close collaboration with the Gloria's lab at MIT. And here is a group of people in my lab. And then we, like many other places, we've been having this Zoom meeting for the lab meeting since, March. And it was just quite painful to, but, we still kind of try to enjoy as much as we can and then, make a progress in our sciences. So I also like to thank my funding sources and would be happy to take any question.
June, thank you so much for an incredibly rigorous presentation of science. We only have a few minutes for questions, so maybe I'll start off, and then there's a couple of questions in the chat that are related to the question I'll ask. But I also wanted to remind all the attendees that you can reach the speakers via the Synchrony app if if your question isn't selected for asking the speakers. And so just very quickly, June, because of time, You know, it's been noted that individuals with ASD have increased inflammatory markers, and there's, you know, elevated TNF and interferon and others. I'm not sure about IL-17, to be honest with you. So how do you reconcile that? Is that a compensatory mechanism, do you think, that there might be increased elevation, or is it just irrelevant because, you know, your work shows that IL-17 is important? You know, how do you sort of come to grips with the increased inflammation that's been observed in ASD?
So I only have my opinion. I don't have any data to support or disprove what I'm thinking. But what I'm thinking is that, you know, those individuals with the heightened inflammation, you know, maybe due to their early exposure to inflammation during pregnancy, you know, so, you know, I know, you know, like we all know that, you know, they're like, you know, the genetic contribution, you know, like 70% of autistic cases maybe due to the genetic contribution, but also, you know, this inflammation or other, you know, maybe gut bacteria induced changes in inflammation also contribute to ASD phenotype. And then, so, you know, based on our work, I think this offspring and also earlier work, you know, by your group and others, you know, suggest that this offspring, you know, born to mothers exposed to inflammation during pregnancy, you know, have heightened inflammation in the, you know, later in their life. So I think, just again, my personal opinion, you know, those individuals with heightened inflammation, you know, may have been exposed to, you know, inflammation, you know, during pregnancy, you know, so that's just my personal opinion.
Sure, and I know this is really at the sort of the frontier of understanding the potential role of the immune system in autism. And again, you know, as I prefaced in the introduction, you know, the mouse models give us targets to study that we can then translate to people. Pramila asks, you know, do you think that there might be any rationale to thinking about Humira or other anti-inflammatories that have already been approved for other disorders, autoimmune and inflammatory and allergic disorders? Any rationale to think that those may be repurposed for autism?
I mean, Humira is an antibody against TNF alpha, you know, and then so I don't, to the best of my knowledge, I don't know whether TNF alpha also, you know, shown to affect this autism-like features in mice. You know, we know that interferon gamma is not important. We know that IL-17, maybe, you know, type 1 interferon and IL-6, you know, clearly play very important role. So, you know, if you're thinking about this prevention, you know, maybe, you know, blocking antibody against those cytokines may be repurposed, you know, to prevent the development of this autism. But again, you know, we don't know whether this finding in animal model, you know, can be translated into human. And I think a lot more study needs to be done.
Yeah, but terrific work, because your research has provided avenues for potential therapeutics both during the prenatal development period in pregnant mothers, but also after diagnosis, potentially after diagnosis as well. So it gives us multiple shots on goal. Thank you very much, June, for
Thank you very much.
your presentation.